Okay, so right, hello, let's get started. So we're going to delve into a multi-stage cloning exercise that should hopefully make you understand all the different things that we have to think about when we're actually doing a cloning. So to start with, we're doing a worked example all the way from the beginning till the end, and then after that you'll be able to do your own using a hexokinase gene from your own model organism. So today we're just going to look at how to clone a gene from one source into Arabidopsis thaliana. And this is the summary of what we're doing. We're getting a hexokinase gene from Arabidopsis thaliana, and we're using PUC18 cloning vector. Then we're subcloning into P. cambia, and then we're going to express again in Arabidopsis thaliana. And you're going to use this sheet in order to put in your answers. And at the end of every session, you submit so that we can give you some feedback as how you're getting along. And you'll also submit the challenges that you have with each stage. And so what I'm going to go through here is showing you what an excellent piece of work would look like, where you've gone into detail explaining all the stages that you've gone through and how you present it at the end. So this is what it looks like, and it's called cloning and expression of wild-type hexokinase in an Arabidopsis mutant. So if you look back here, we said we're getting the hexokinase from Arabidopsis, and then we're expressing it again in Arabidopsis thaliana. So what would cause such a situation to be needed? So the problem we're going to have today is that we've got this process called glycolysis that you know very well, and we're looking at this one enzyme hexokinase, and we're asking ourselves, what would happen if you mutated it? What would happen if this enzyme was somehow not working the way it's supposed to be? It was mutated or knocked out altogether. So what would happen? And if we look at that particular enzyme, it looks like this. If you go into the protein database on NCBI and you look up Arabidopsis hexokinase, this is what it looks like. But before you can really work with it, you need to know what kind of sequence you need to get from it. And it needs to be the wild type. So is it the genomic sequence? Is it the mRNA sequence? Is it the protein sequence that you're interested in? Let's delve a bit deeper and see. So what happens if you do mutate this particular gene? Well, in this paper, they explain the kind of phenotypes that they saw when they mutated this gene in Arabidopsis, and it wasn't only phenotypes of nutrition, because this is a nutrition pathway, right? It's how the organism takes glucose and makes it into energy in terms of ATP and reducing power. So what they noted is that not only was there nutrient issues, there was also issues with detecting light and issues with hormone signaling. So this should tell you something, that even when you learn about some of these pathways, just because an enzyme does this, it doesn't mean it doesn't do other things as well. So it looks like if you were to knock out this gene, knock out the expression of this protein here, not only do you affect the nutrient cycling in this organism, but you also affect its ability to look at the hormones around it and signal, and also to respond to light, which is very interesting. So that should make you understand that these pathways are usually quite interrelated, and proteins do more than one thing. So what does Arabidopsis look like? It looks like this, a little weed. I'm sure you've seen some growing in the cracks of pavements. And it has this basic form where it's got the flowers, little white flowers with the calyx where the seeds will be. And then it's got these rosette leaves and some roots as well. So this is what it looks like. And it's in the cabbage family, the brassicas. And so it goes through that same bolting, like the way rape does, or cabbage, or Chinese cabbage, and so on. So this is how it looks in the wild. And this is how it looks if you knock out the hexokinase gene. So in this plant that's been knocked out, this is the wild type. So this is how it naturally looks like that in the lab. And this is what happens if you get that one gene and you knock it out, and this is how it looks. So you might want to just remind yourself of the difference between reverse and forward genetics. There are times when we start with a genotype. In this case, we know this is the gene involved. We've knocked it out. This is the phenotype we see. There are times where you start with a phenotype, where you just do like a random mutant screen and you don't know what you've hit, and then you start looking for phenotypes, and then you go figure out what the genes are that cause those phenotypes. So in this example, we're starting with an actual known gene. We know that this phenotype is caused by a gene we know, which is the Arabidopsis hexokinase gene. You know that if you knock it out, this is what it looks like. But you should be able to compare the difference between going forward or backwards in these types of genetics and give examples of it too. So the real question we're asking in this exercise is, how can we rescue this phenotype? By rescue, I mean, how do we get this sick plant to be well again? How do we get this sick plant to be well again? And in genetics, we call that rescue. We rescue the phenotype. So the most straightforward way is give the plant what it needs. The one thing that is missing is a correct copy of this gene, right? So if we were able to give it the correct copy, we should, in theory, be able to rescue this phenotype and make it look like this. So the question is, how do we go about that? And that's what this cloning exercise is all about. So let's spend the rest of this project learning how to do this. Our objective, we want to clone the Arabidopsis thaliana hexokinase 1 gene from a healthy Arabidopsis wild-type plant, Columbia background, into Arabidopsis thaliana mutant, which is this one, which is also known as Gen 2, and that is what was written in this paper here. So they called it Gen 2 as well, the people who were working on it. But we'll be referring to it as Arabidopsis thaliana hexokinase gene 1. And so if we do that, it means we'll be able to learn more about the things that it affects: the glucose metabolism, the sensing of light and energy, and plant growth regulation through hormones. Now, you might have noticed that I've written this word in a manner of different ways. Sometimes it's italic, sometimes it's straight up, sometimes it's small letters, other times it's big letters. So I'd like you to look at this paper, or rather this document, which goes through the nomenclature that we use to be able to differentiate between gene names, protein names, wild type, and mutant alleles. So you go through that. And also, if you do a mutagenesis of some kind, there's a way of writing down to show anybody who's reading exactly what mutation you did and what that mutation means, how the organism has changed, whether a codon has changed, or whether this is a point mutation, or different types of things. So you can have a look at this document to be able to refresh yourselves on that. So back to the mission at hand. We want a wild-type gene, and I've looked it up. The hexokinase gene in Arabidopsis is AT4G29130. So if I do a search for Arabidopsis thaliana hexokinase gene, this is the gene that I'm going to get. And we've chosen it because it's a well-characterized gene that encodes a well-characterized enzyme. And we're going to use the E. coli cells for intermediate hosts. That's because they're easy for cloning and amplification of our gene. And our final host is going to be the Arabidopsis thaliana mutant. And we're using Arabidopsis thaliana because it's a model organism, and its methods for transformation are very straightforward. We know how it works. So in order to get more out of this, I would like you to fill in the data about your own model organism and its hexokinase homologue in the shared Google Drive. You can ask your class rep for the link. And for that, it's just it's going to look like this. So when you get the shared Google Drive, it's this one, Biotech Year 3, 2026, and in there's two folders. So there's the BT325 folder and the BT310 folder. And in the BT310 folder, that's where you'll find some more material support to support this work. In the BT325 folder, that's where you'll find the sheet that you need to fill in your own organism into. So you've done this work already, but you can just augment it by doing the little things I've put at the bottom. This is the example for Arabidopsis thaliana. So you can see down here, the name is written. You put your student name and your computer number, and then down here you've got all the things that we're interested in. So what's the name of the organism, what's the domain, the ploidy, the chromosome, all this stuff. You write it all down. Then over here you put the links to the picture of the organism, a link to the taxonomy page on NCBI, and so on. And then over here, this is the bit which links with the work that you're doing right now. In the genes section, we want the hexokinase gene and annotation number, or rather accession number, from, I'll put here accession number. Don't know what I was thinking. Accession number. Yeah. So you need the one for the gene, the one for the protein, and also when you've gotten this, can't spell today, when you've gotten this, you want to see which chromosome is your actual gene on, and what is the locus on that chromosome, and then get the hexokinase gene sequence and the hexokinase protein sequence. And then down here you put in the zero cutters, so the restriction enzymes that don't cut at all, the ones that cut once and the ones that cut twice. And then here you put in the primers for amplification of your hexokinase from your organism and for cloning. And you'll see in a minute how we do that. So if your organism is Daniel Aereo, you basically copy this and then put it here and fill it in with the information for yours. If you're Saccharomyces, if you're Escherichia coli, and so on, here just add another tab, add another sheet for your new one, if I haven't written it down, because you've got many more, isn't it? So by the time we're done, we should have this sheet about all the organisms in this Google sheet. All right, let's go back to this. So you can ask the rep for the link to that Google Drive, and you can look around and take the things you need from there. So let's look at the strategy we're going to use. Step one is we're going to use a cloning vector, and this is one you've already met before, PUC18. Why? Because it's high copy number, ampicillin resistant, and it's got a multiple cloning site that we can use in order to do the cloning. Then you can get that sequence from AddGene. Remember where AddGene is. You remember this? So go to AddGene, in the search bar, put in your the PUC18 and get the sequence. If you're not logged in, you won't be able to get the sequence. So then we're going to need the expression vector. So for that, we're going to use the P. cambia family of plasmids, because these are plant compatible and they contain a 35S promoter right here somewhere. There it is. This promoter, which allows the expression of whatever insert you're going to put there, which in our case is our unmutated hexokinase gene, because we're going to take this plasmid with our unmutated hexokinase gene, the wild type one, and we're going to put it into the mutant plant, and then the next generation you should see the plant is rescued, so it's healed from its mutation because of what we've put in there. And the selectable marker for this one is hygromycin or kanamycin, and you can see exactly where they are and which direction they are. And you can get the sequence for that from AddGene and put it into your documents as well. And the genetic engineering tools we're going to use, we're going to need some restriction enzymes such as ECOR1 or HIND3, and we'll see in a minute how we'll choose them. And the reason for this is because we need to cut the vector and we have to put in our insert. We're going to need DNA ligase to join the fragments together. We're going to have to use PCR in order to amplify our gene. We're going to need competent cells in order to put our plasmid with insert into those cells so that they can be transformed. And then we're going to need antibiotic resistance or antibiotics, and we're going to select using the antibiotics for the clones which are actually the ones that we want. So we want the cells which have picked up the DNA, and if they've picked up the DNA, it means they'll have the genes for resistance to whatever antibiotic we're going to put in the media. So that way we can kill off all the ones that didn't pick up the plasmid, and only the ones that picked up the plasmid will be able to survive. So a summary is, we're going to get our hexokinase gene, we're going to amplify it using PCR, clone it into PUC for propagation, then subclone it into pCambia for plant expression. So that's what you're going to do in the first session. You're going to do the whole thinking behind that. And then in the second session, we're going to now look at the cloning techniques. So we're going to go a bit deeper. What are we going to need? We're going to need the wild type Arabidopsis plant, and we're going to need to get the DNA from that. So extract DNA to provide unmutated Arabidopsis thaliana hexokinase gene 1. But wait, it's a bit more complicated than that, because there's a difference between the types of DNA that I can actually be using. I actually need cDNA, a copy of the gene, because of the reasons outlined below. So we've got two reasons for that. One, if you look at the stability of RNA and DNA, it's not the same. You can just check this table and see that RNA is really not that stable. DNA is much more stable, and that's why it's easier to work with. So that's one reason we do that. The other reason is here. If we look at genomic DNA versus cDNA for expression, with genomic DNA, you've got this issue of introns, so it needs splicing. That means that the expression efficiency is not so high, and it's variable, and it can be unpredictable. Whereas with cDNA, you don't have any introns present, it doesn't require splicing, and it's much more efficient when it comes to expression, and you can predict much more highly what's going to happen. And that's because you've got all these elements that you've put into the plasmids that you're using. So remember here, we had our promoter. If this promoter is here, and then you've cloned in your insert there, you can predict how much expression there's going to be. Whereas if you're using the native setup, so the promoter from Arabidopsis itself, and the way it just occurs in its own natural state, it's expressed much less. So this one is actually from a virus, cauliflower virus, mosaic virus, and you know how viruses are. So it actually ramps up the production of whatever it is you put in front of it. So because you're using this cloning system, you're able to get more copies than you would if you're just using the regular Arabidopsis promoter and gene. So that's why we've got to actually do a bit more work. We don't clone the genomic DNA, we clone the cDNA. And if you read here, there's the whole protocol of how you do it, where you start with your DNA and do extract your RNA and do your RT-PCR, and then use that for your actual final PCR and so on. Now, I just wanted to look more into that so you really understand what I'm talking about. In front of you here, this is a piece of genomic DNA. And for you to understand the difference in genomic DNA, and this is your cDNA, you can see that the size is different, but there's also a very fundamental difference between them. As we said already, one has introns, the other one doesn't. Let's see what that means. If we go over here, this is our Arabidopsis thaliana karyotype, okay? This is it's got 1, 2, 3, 4, 5 chromosomes, and this is how big each chromosome is. They're not the same size. Then this is the mitochondrial chromosome, and this is the plastid chromosome. So if you go to plants ensemble.org and look for Arabidopsis thaliana, this is a database for Arabidopsis, but it's for all model plants. So you find the Medicago and all these other ones there too, and they've organized this information like this. So if we start here and we said this is how the chromosomes are. If I looked at my number, you remember the number of my gene? If we looked at this, the gene was called this, right? AT4G 29130. That means it's AT, Arabidopsis, 4G, 4 chromosome. And if I look on this same 4th chromosome, and I look at the position where this gene is, now this is doing this, fantastic. So this right here, usually the chromosome comes up here. There it is. So this is the 4th chromosome. So you saw that I came from here. If I click this one and I'm looking for my gene and I zoom in on it, this is the 4th chromosome and it's around there in terms of position. And if I zoom in a bit more, I'm going to see my gene. So this is my hexokinase gene, this one. AT4G29130, hexokinase 1. So this is the gene that we're busy discussing. And I want you to pay attention to the structure that is there. So if you can see how many mega bases of DNA we're at. If we come back here, you have to open it for yourself, but there's a ruler down here which tells you like how many mega bases of DNA this whole length is. But you can see here that we're already at 14 mega bases, 14,000 basically. It's a lot, right? So we look at the structure here. This is the gene that we're discussing, this one right here. And you can see it starts with this clear box, then a filled in box, then these things like that. So what it is untranslated region, exon, intron, exon, intron, exon, intron, exon, intron, exon, intron, exon, intron, exon, untranslated region. But wait, there's more. If you look at the arrows, you can see that there's something which is not the same, right? And if we look up here at this same gene of ours, it tells us that this gene has 7 exons and it's found on the reverse strand. Okay, meaning if we look on this map, if this is the normal direction, it's found on this direction. And you can see here these are on the leading strand, that's why the arrow is pointing this way. These are on the reverse strand, which is why the arrow is pointing that way. So this is the beginning. The ATG is here. It goes this way until the stop codon and then the 3' UTR. So you have to bear that in mind. But that's how it is with the genes. Some genes can go this way, some genes can go that way. And you can see it more clearly here. There's your hexokinase gene going this way, and then there's a bunch of other genes going that way, and so you have to pay attention to that when you're working with your DNA. Now, if we go back here, I want you to understand the difference between this genomic DNA and the cDNA. So if we look here, where is that? Now how am I gonna find that? Hope it's this one, but you can just make the image a bit bigger for yourself. So what I've got here is showing you this story right here, this one, and you can get that by clicking the link. And this document, actually, it's supposed to be a Word document. I just have it here as a PDF to show you, but for you to do any kind of analysis, you need the Word document because you want to be able to really use the Word tools. So I'll show you from the Word document that I have. So if you look at this, Here's my genomic DNA, and that is that area of the chromosome that we highlighted. So it's a big chunk. But what is going on in here? So if we look a bit more carefully, this is where the coding region is, the ATG and the TAA. Now, if we look at this one, it shows us the actual intron exon structure, which is why I want you to look at it. So you can follow this link. And it will show you much more in depth the intron exon structure, so you can understand the difference in the genomic and the cDNA. Let's see. Takes a while. So while it's taking a while, it's quite simple. So this was the untranslated region we talked about, and we saw from the right up there, we've got the exons and the introns. So there's the first intron, the second intron, the third intron, the fourth intron, and then it gives you the pieces which are actually going to be found inside the final protein. So it looks like that. Untranslated region, this is the bit that will be in the protein, that's the intron, the bit that will be in the protein, and so on and so on and so on.